sit down in your desk. Okay. Okay. I believe everyone has nice lunch or dinner, so let's start the last session today. There are 3 speakers in this last session, and the first speaker is Shlomi Reuveni, and he will talk about restart, the science of starting anew. So let's welcome the speaker.
Thank you very much. I'm just making sure that everybody can hear me okay, see the screen and see my cursor.
Yes, we can hear you. Yeah.
Okay, so let me start by thanking the organizers for their kind invitation. It really is a great pleasure to be with you online, but still. Before I start my talk, I just want to give you a brief overview of the Reuveni group and what we do. First, we're located at the School of Chemistry of Tel Aviv University, which is in Israel. And we are a theory group, so we do basic theoretical research in the mathematical and natural sciences. This is fairly broad, but more specifically, we're interested in randomness. And so we study complex systems that are governed by statistical laws and random events. You're going to hear about that a lot today in my talk, which is going to be focused on resetting and applications. And I will emphasize applications in biophysics. Another thing that we do, very much related to randomness, is we develop theory for single X science. And what is the X? The X could be a single molecule, single cell, etc. Whenever you go from the level of the ensemble to the level of the single entity, randomness plays a role. And we're interested in developing theoretical tools to better describe and understand such cases. In particular, we're interested in chemical reactions that are going on at the single molecule level. And here you can see a little cartoon from a recent paper that we published in PRL, in which we investigated gating, this property of a molecule to switch between a reactive and a non-reactive state stochastically, and how these transitions affect the reaction time. So I will not talk about this today specifically, but you'll hear about other aspects of single molecule theory that we're developing. Another thing that we do in the group is try and understand how evolutionary optimization shaped the molecules of life. And one such molecule is the ribosome, the machine that makes all the proteins in our bodies, synthesizes all the proteins that we have in our bodies. And recently, we were able to show that the composition of this machine, it's made out of RNA and protein in a very specific 1:2 ratio. We found out that this is not a coincidence. Rather, this is a result of an optimization. Optimization for what? For the speed at which cells grow and divide. So I'm not going to talk about this today, but if you're interested in such ideas, maybe have a look at our papers or contact me after. And now for my talk, and I'm going to focus on restart, the emerging science of starting anew. So this has really been an emerging topic in physics, with applications to statistical physics, biological physics, chemical physics, and also in other fields like computer science. And what I'll try and do in my talk today is pique your curiosity regarding resetting, stochastic resetting. Why is it important? Why is it interesting to think about processes that are being stopped and started anew from time to time? I'll do that initially just to build ideas using very simple toy models, and then we will go on and consider applications to situations where resetting is actually baked into the cake. It's already part of the system. And one application that we will consider in the context of biophysics is going to be enzymatic reactions at the single molecule level. And more generally, Michaelis-Menten type of reactions. And these could be things that are not enzymatic catalysis per se, but many other things that are going on in the cell. And another application that I will mention of resetting theory is to search processes. These could be molecular search processes, like maybe facilitated diffusion, or search processes on the organismal level, where an animal is foraging for food and comes back to its home from time to time. And this is also a type of more sophisticated resetting that we can account for and within the framework of the theory. And you'll see how everything is weaved together. But let me start with two very simple examples of what do I mean by resetting, and then also why is it interesting, at least one aspect of resetting study that is interesting to consider. I want to think about processes that are being stopped and started anew from time to time stochastically. And for that, I will take the simplest process possible. Let's think about motion with constant velocity. So I have a particle here and I have a target, and the distance between the two is L. And at time 0, I'm going to let the particle move towards the target at a constant velocity V. And then I will ask you, how much time does it take the particle to reach the target? What is the first passage time of the particle to the target? Of course, it's just L over V. Now let's consider the same thing, but with resetting. So I want to now reset the process at some constant rate R. And what this means is that I will have a certain probability, R times delta T, for every infinitesimal time interval delta T. For what? For me to take the particle and place it back at the origin, reset its motion. So now I can have situations like that where the particle starts moving, but then resetting occurs at some random time. I place the particle back at the origin and let it go again, try again, until it is able to hit the target. And if I'll ask you now how much time it takes the particle to reach the target, you should say, well, it's no longer a deterministic process. There is some distribution that will govern the first passage time. However, without doing any fancy calculations, you'll agree with me that on average, it will take the particle more time to hit the target with resetting than without, regardless of what this resetting rate is. And why is that? Because, the last trial in which the particle was able to reach the target uninterruptedly took L over V units of time. And then prior to that, maybe, we had 1, 2, 3, 4, I don't know how many events in which the particle started moving towards the target, but was interrupted by resetting, and it had to try again. And these failed attempts took time, and this time must be averaged in. And of course, it will increase the mean first passage time of the particle to the target. And now you could say, okay, sure, I can generalize from here pretty easily. Take a process, any process that you can imagine, stop it and start it anew from time to time. All you're going to get by doing that is hinder its completion. Stopping something in the middle, just to start it all over again, this is a waste of time. It's going to hinder completion. Any process doesn't have to be this toy model that I'm looking at. And in fact, we all have this intuition deep inside, but this intuition turns out to be fundamentally flawed. It's completely wrong. And let me give you one example of a process where it breaks down. So let's think about a similar setup, but here I will have a particle undergoing diffusion or random walk until it reaches a target, and the distance again is going to be L. The diffusion is going to be governed by some diffusion coefficient D. And I can ask the same question, how much time does it take such a particle to reach the target? Of course, there is now a distribution, a first passage time distribution, and it is a classical result in diffusion and stochastic processes to compute the shape of this distribution. It looks exactly like that. This is the analytical formula. What I can tell you about it is that it's normalized to 1, which means that if you give the particle enough time, it will reach the target. However, it is also extremely heavy-tailed. And so at very long times, this distribution falls like 1 over t to the 3/2. And so if you multiply by t and integrate in order to get the average time it takes the particle to reach the target, you'll find out that you're integrating over 1 over the square root of t, and this diverges. So the mean first passage time of a diffusing particle to a target actually diverges. Okay, this is all classical results, which were known for a long time. But what happens now if I incorporate restart into this process? So again, I can restart it with some rate R. And the first to consider such model were Evans and Majumdar in a PRL paper that was published in 2011. And what they discovered, among many other things, is that when you introduce resetting, no matter what the resetting rate is, you will take the mean first passage time, which previously diverged, and make it finite. So let's think about it for a minute. We started with a process that on average took an infinite amount of time to complete. And just by stopping it and starting it anew from time to time, we made it complete in finite time on average. This is a huge improvement, and of course, completely different from the type of effect that we saw for the deterministic or ballistic motion. And of course, you can now think about this more generally. You can think in general about first passage under restart. So maybe you have a group of searchers that are looking for some target, maybe it's a missing person, and then bad weather or nightfall will force these searchers to go back to base. And by the time they start their search anew, the missing person has already relocated. It's a completely new search process. This is first passage on the restart. Maybe you have a computer algorithm that stochastically scans a tree of possibilities in search of an answer to some mathematical problem. But then, randomness could take this algorithm down the wrong path. And maybe you would like to code into your algorithm resetting such that it will have another chance, a second chance of finding the solution to the problem that you're considering. And in fact, computer scientists do this all the time when they're dealing with stochastic algorithms that can get stuck, like I illustrated here. Maybe you have some molecule that was prepared at an excited state and then it decays to a low energy state, but this low energy state is not the low energy state that you were hoping to get. It's not the product. So maybe by a zap of laser or mechanical manipulation, you can reset the configuration or re-excite this molecule. And then, maybe now you're going to have the product that you wanted to get. All these processes fall under the broad umbrella of first passage under restart. You have a starting point, you have a finish point, you have some first passage process that takes you from start to finish. And then on top of that, you also have resetting. And what is the million-dollar question that we would like to answer? What is the effect of resetting? So if I give you a process and I tell you, okay, let's now try to reset it. What is going to be the effect of that? Is the process going to finish faster or slower? And moreover, what do I need to tell you in order for you to be able to answer this question? Do I need to tell you all the details about this process, or is it enough that I will tell you just a handful of details that are important? Is the answer highly dependent on the details of what you're looking at, or is the answer universal? It turns out that the answer is indeed universal, and this is what we discovered in these set of papers. I will not prove it. I just want to today, but I just want to describe it and then also discuss its meaning. So it turns out that all you need to know in order to decide if resetting will hinder or expedite the completion of a process is the CV, or the coefficient of variation of the distribution of completion times. What is the CV? The CV is just a dimensionless measure of dispersion. It's just the standard deviation of the completion time distribution over the mean. It's a measure of relative fluctuations around the mean. And it turns out that if the CV of the completion time distribution of your process is larger than 1, in a sense, your distribution is slightly even wider than the exponential distribution that has a CV which is exactly 1. If this is the case, then resetting, or the introduction of resetting at least to first order, will lower the mean first passage time. Otherwise, if you have a narrow distribution, CV smaller than 1, then the introduction of resetting will hinder completion. I want to illustrate this result by considering an example that interpolates between the two extremes that we started with, pure drift and pure diffusion. So let's think about a particle undergoing Brownian motion, but with some drift. So this particle starts here, and there's an absorbing boundary at L, And the particle drifts towards the boundary at some velocity v, but it's also diffusing. And so here are the trajectories, and I'm restarting them from time to time until the particle reaches the absorbing boundary. And I'm interested in the average time, in the mean time it takes me to get to the boundary in the presence of resetting. What is interesting is that this, or the profile is actually going to depend on how strong the drift velocity is. So here I'm plotting the mean first passage time of the particle to the target as a function of the resetting rate. And what you can see is that, let's consider this case first, the case where v, the drift velocity, is 0. This is just pure diffusion. What you can see is that without resetting, we're back basically to the problem of a particle diffusing and needs to hit a target. On average, an infinite time will be taken for the particle to do that. So you see the divergence here. But the minute you introduce resetting, the mean first passage time becomes finite, and it minimizes, it attains a minima at some critical resetting rate. And then it goes up again because, if you're resetting too frequently, you're not really giving the particle enough time to explore its surroundings. And this non-monotonic trend in which resetting first reduces the mean first passage time and only then you go back up continues, and as long as the drift velocity is not too strong. Once you cross a certain threshold, you see a phase transition, okay? You see a phase transition between this non-monotonic behavior to a monotonic behavior. And we expect this phase transition because you know that for high drift velocities, we're virtually in this situation where we have a particle moving and just little fluctuations around this deterministic motion. And we know that over there, resetting only hinders completion. So of course, I can do this by changing the drift velocity, but I can also change the magnitude of the diffusion coefficient or the distance between the particle and the target. And the physical intuition really tells us that there has to be a way to compact all these things into a single dimensionless parameter that will tell me exactly where is the transition point. The transition point between resetting helping or reducing the magnitude of the mean first passage time and vice versa. And really, for drift diffusion processes, we know what the natural thing to take. It's the Peclet number. It's just the ratio between the rate of advection and the rate of diffusion. So this is the typical timescale for advection. This is the typical timescale for diffusion. You take the inverse, you get the Peclet number. It's just LV by 2D, where L is the distance from the target, V is the drift velocity. If it's high, the Peclet is high. D is the diffusion coefficient, and if it's high, the Peclet is low. So this dimensionless number quantifies the competition between drift and diffusion, and we expect that the transition point should be uniquely determined by the Peclet number. On the other hand, we have a general theory, I just told you, that tells us that the transition point is determined by the CV. All you need to do is look at the distribution of first passage times, take the standard deviation over the mean, and see if this is larger than 1 or smaller than 1. If this is larger than 1, resetting is going to be worthwhile because it's going to decrease the mean first passage time and expedite the completion of your process. And really, when we plot these distributions, you see there's some connection between the width of the distribution and the Peclet. For high Peclet numbers in the strong drift regime, the distributions are narrow. And for low Peclet numbers, they're much wider. And in fact, we know analytically how to compute the distribution of first passage times for this problem. And what you can do is then compute the standard deviation, compute the mean, and form the CV. It turns out that in this problem, the CV is exactly 1 over the square root of the Peclet number. And so CV larger than 1 is Peclet smaller than 1 and vice versa. And the physical intuition really sits very well with the general theory. The importance of this general theory is that these conclusions can be extended to situations where we have no notion of the Peclet number. It doesn't have to be drift diffusion. It can be your favorite process that has some distribution of completion times, and looking at the CV will give you the answer of what would be the effect of resetting for such a process. Now, I want to move on and discuss some applications to biological physics. And for that, I want us to know that contrast to the toy models that I started with, restart must take time in the real world. So, if you reset the process, you're not really teleporting a particle back to its origin, or if it's a computer algorithm, you can restart it in 0 time. There is some overhead that will have to be taken into account. And so what you really want to consider is first passage on the restart with time overheads, because every time you reset, you're going to have to pay some time. Now, this could be a mathematical trick to extend or generalize the model to account for such situations, or some mathematical curiosity. But in fact, there is strong motivation to do that because there is a 1-to-1 mapping between first passage under restart with time overheads and the model of the Michaelis-Menten reaction scheme at the single molecule level, which is a model that we use in order to describe enzymatic catalysis. And so a protein binds and unbinds its substrate. And when they're bound, they form a complex and you can get your product via catalysis, which to a change of labels can be viewed as the first passage process in the context of enzymatic catalysis. Unbinding is of course equivalent to resetting. And then there is a time overhead that you need to pay, because if the enzyme and the substrate unbind, you're going to need to wait some time until the enzyme binds another substrate. And so you see here that if we want to better understand first passage under restart with time overheads, we should better understand enzymatic catalysis at the single molecule level and vice versa, because they're completely equivalent. And moreover, we can learn some new things about enzymatic catalysis by considering this thing. And this is what I want to do, show you now what these things are. But before, I want to say that, this reaction scheme, the Michaelis-Menten reaction scheme, we use it to describe the action of enzymes, but we actually use it also to describe many other things that are going on in the cell, like transcription, the translation, the work of linear motors, et cetera, et cetera. Let me give you 2 examples that fall under the Michaelis-Menten reaction scheme, or first passage under restart with time overheads, that maybe you didn't thought about before. One is facilitated diffusion. So maybe I have some DNA and there's a target site on the DNA. And then, there's an enzyme or a transcription factor that binds the DNA and tries to find this target, okay? Then, it doesn't have to be a chemical process that is being catalyzed. You can think about any process that goes on, any first passage process. In this case, the catalytic process is just the search process itself, the enzyme searching for the target. And then you also have binding and unbinding, which are equivalent to resetting and the time overhead. So this is one situation in which, still we have first passage under resetting with time overheads. Another case is chaperone-assisted protein folding. So an unfolded protein, after it was synthesized, will fold to its native structure, but sometimes you get a folded protein, but sometimes you'll get stuck in a local minima of the energy landscape. And then nature invented these beautiful machines called chaperones. And what they do is they come to these folded proteins and they unravel them. They basically restart mechanically the folding process and give the protein another chance to fold. So this is another example of resetting that is baked into the cake, basically, in biophysical systems. And what we want to understand is what is the effect of resetting in such systems. So here's the classical textbook treatment of resetting or unbinding in the Michaelis-Menten reaction scheme. What you do is you take this scheme and you put rates on the arrows. And this is basically amounting to assuming that all these processes are exponential. And then you can plot the rate of product formation, the number of products that you get per unit time, as a function of the unbinding rate. And what you'll get is a monotonically decreasing curve, which is not surprising. The higher the unbinding rate, the higher the backflow, the less products you get per unit time. However, we now know from single molecule measurements that this step at least need not be exponential. You can have non-exponential distributions of the catalysis time. And the question is, does it matter? And if so, how does it matter? And it turns out that similar to the case of, so what about non-exponential distributions and of catalysis times, and what is the effect of these things? And it turns out that similar to what we saw before, depending on the width of the catalysis time distribution, you could have different behavioral profiles of turnover versus unbinding. So again, what you need to do is form the ratio between the standard deviation and the mean of the catalysis time distribution. This is called the CV again, or the coefficient of variation. If it is smaller than a certain threshold value, which I will mention in a second, what you're going to get is a monotonically decreasing profile of turnover versus unbinding. However, if it is larger than this threshold value, you have large fluctuations, and then what you could get by increasing the unbinding rate, an increase rather than a decrease of the turnover rate. So this is a non-classical effect that gets bigger and bigger the larger the fluctuations around the mean become. And so what is this critical value? Previously it was 1. We needed to see if CV is larger or smaller than 1. Now, because we have a penalty, every time we unbind, we need to wait some time for binding again. Now, this penalty has to be taken into account. And it turns out that it comes rather generally in this form. You have 1 plus, and then you have twice the ratio between the mean binding time, or the mean overhead that you need to pay, but normalized by the mean catalysis time. So CV is dimensionless. This side also needs to be dimensionless, and the penalty is actually weighted by the mean catalysis time. What is interesting is that this extra term here actually could be made as small as you want, arbitrarily small, by taking the substrate concentration to be high. Why? Because if you have a lot of substrate, binding the enzyme, binding another substrate will actually take very little time. The binding time is inversely proportional to the substrate concentration. And so in this limit, you're going to need CV to be larger than 1 and nothing else. So this is a different look at the phase space. Here I'm plotting substrate concentration, and here I'm plotting the coefficient of variation of the catalysis time distribution. And what you can see is that once your distribution, the catalysis time distribution, is even slightly wider than the exponential distribution in the CV sense, then there will always be a substrate concentration high enough for you to see this non-classical effect, in which unbinding actually increases the rate of product formation. There is a twist to this story, which involves enzymatic inhibition. Inhibitors are third-party agents that come and bind the enzyme. And when they bind the enzyme, they block product formation. So this is what they do. There are all sorts of different types of inhibitors. And here's an example of one type of inhibition mode that is called uncompetitive inhibition. This inhibitor binds the enzyme substrate complex to form an enzyme substrate inhibitor complex. And when you have something like that, you see that you cannot get to the product directly from here. You first need to unbind the inhibitor. Okay, but similar to regular unbinding, which takes you to a state, a free state from which you cannot get directly to the product, this binding of an inhibitor may also work as a resetting agent via a third party. And if you take that into account, and in addition, take into account the fact that the catalysis time distribution can be non-exponential, and in particular have large fluctuations around the mean, what you will get is, in addition to the regular or classical situations where you have more inhibitor and then the rate of catalysis goes down, what you will find are situations in which you increase the concentration of the inhibitor and the rate at which you accumulate product goes up. So stochastic fluctuations at the single molecule level can make inadvertently inhibitors act as activators, and we should keep that in mind because we use inhibitors all the time in all sorts of different situations. Antibiotics is one example. So what this means is that we need a new theory of inhibition, a new theory of single molecule inhibition, and this was formulated in this paper here, which I invite you all to read. Now I want to finish by talking a little bit about restart in the lab. And here's a video from the first experimental realization of diffusion with stochastic resetting. And this was done in the lab of Yael Roichman by very talented students, Ophir Tal-Friedman and Amandeep, obviously also with the talented students from our group, Arnav Pal, who has now started a new position. What this is showing you is the following thing. You have a particle diffusing, and from time to time, Yael comes to this particle and grabs it with optical tweezers and resets its position. What you can see is that resetting takes time, but there's an additional complication. It's not only that resetting takes time, it actually matters where you grab the particle. If it's far away, it will take you more time to bring it back. If it's close, it will be much faster. So resetting not only takes time, this time is space-time. There's some space-time coupling, which tells you that this time actually depends on the state of your particle or its position here, which defines its state. This is important. This is important when, or this complication is important when thinking about search with home returns that we see in nature. And for this, let me quote Darwin, who in his famous book in 1861 wrote that it may be here remarked that most animals and plants keep to their proper homes and do not needlessly wander about. We see this even with migratory birds, which almost always return to the same spot. So Darwin observed that animals have homes, okay, dens, nests, and so on and so forth. And you know, we all recognize the benefits of having a home, but what we came and asked is maybe there's a benefit, additional benefits of having a home besides warmth and shelter and all the usual stuff. Maybe having a home actually will help an animal locate food and other resources much more frequently than a situation where it does not have a home. So I want to think about free-range search and compare it to home-range search. So maybe, you know, I'm starting here and I have the butterfly that starts at some position, and then it searches and searches and searches until it finds a flower. And in contrast, I have the bee, which you know, searches, and if it doesn't find it, it returns home, and then it searches once more, and if it doesn't find the flower, it returns home, and then it tries again. And the question is, what is a better strategy of search? Who will find the flower first, or a flower first, and in what situations? Can we divide the phase space very generally, like we did for the effect of resetting, and tell you, you know, in these situations, it's better to do free range search, don't come back home. In these situations, it's actually better to do home range search. You should come back from time to time, and this will actually help you get or find the flower faster. So you have these situations of search. If you find, great. If not, you need to return. The time it takes you to return depends on where you are at the moment that you decided to return. And on top of that, you can also spend additional time at home. And this all seems wasteful, right? Because you're, instead of searching for something, you're either returning or waiting. And it seems that free range search may be a winning strategy in any case here, but it's not the case. It's not the case. And again, what turns out to be the case is that the CV, or the coefficient of variation, in the time it takes you to locate a flower in the absence of resetting, will determine if it is better to do free range search or home range search. And in particular, search with home returns will provide advantage in conditions of high uncertainty. In conditions where the CV is larger than 1 plus the same penalty that we had before, which comes from time you spend at home, but then there's an additional similar penalty that comes from basically the ratio of the time it takes you to return, the average time it takes you to return over the time it takes you to locate the target in the absence of resetting. So these are two penalties that you have to consider. But if fluctuations are large enough, it's going to be worth it to come back home from time to time and restart the search. You're going to find resources faster in that way. And this is, again, extremely general, does not depend on the details of how you search. All you need to look at is the CV of the first passage time distribution in the absence of resetting. I want to end by mentioning a recent paper of ours. So maybe you heard this talk and you're interested in knowing more. I didn't do any calculations today, but perhaps you want to start, get some easy intro to stochastic resetting. Where should you go? For that, I just want to mention this connection that we recently found between stochastic resetting and the inspection paradox, or Feller's paradox. So Feller's paradox from probability theory is the following paradox. You're looking at a bus station, and buses are coming, one by one, and there's some time between bus arrivals, stochastic time between bus arrivals, and some average time between bus arrivals. If the fluctuations in this time are large, it turns out that if you arrive to the station at some random time, okay? If you'll arrive to the station at some random time, you might end up waiting on average longer than the average time between bus arrivals. This is counterintuitive because you come after a bus has already departed, and yet you need to wait more. How is this possible? But it turns out that here again, CV equals 1 is the sweet spot. If fluctuations are larger than that, you're going to wait longer. And it's not coincidental. There's in fact, you can understand all the results that I showed you today with the aid of the inspection paradox. And we've written a paper on that, the inspection paradox in stochastic resetting, basically telling you what is the mapping and also surveying the inspection paradox and its applications. The upside of this approach, as opposed to previous papers that we and others published on this topic, is that it only requires elementary mathematics. Almost high school mathematics is required and nothing more. Almost only high school mathematics and nothing more. Maybe first year university, but it's very simple if you go from this approach. Okay, with this, I would end. I would like to thank you again for inviting me to speak. I want to thank our funding agencies, Azrieli Foundation, ERC, Israel Science Foundation, group members that have been involved in research, some of the research that I showed, some of the research that I didn't review, but mentioned in the beginning, and collaborators. And with this, I would be very happy to take questions. Thank you.
Thank you very much. Is there any questions? Yeah. First. Do we have mic?
Yeah. So somebody raised his hand.
Yeah. On site, there are 2 people raising hand. Yeah. Go ahead first.
Thank you for the great talk. I really enjoyed it. But
can you please speak up? I can barely hear. If you can speak up. Okay, going to the microphone.
Yeah, so I still didn't get why this concept of first passage under reset would be useful, because the fact that you can reset something means you know the exact location of that object which is diffusing or undergoing first passage. And you also need to know where the origin is, where the starting point is, right? So it's just a very simple process, right? If you know where the particle is now, if it's close to the destination, then it hurts to reset back to the origin. If it's further away from, it's in the opposite direction, so the distance is further away than from the origin, then it of course helps to do the resetting. So why would anyone want to know about the CV and the mean first passage time of this process?
Yeah.
We already know exactly where the particle is.
Yeah. So that's a great question. You're basically asking, if I know where the particle is, right? I can basically decide if I want to reset it or not, if it's closer to the target but imagine that you don't know. Imagine that you don't know. In these toy models, okay, we're just playing around with the idea that we do some blind resetting. I don't reset based on the information. I just maybe I have a mechanism to reset the motion of this particle. For example, imagine that I'm not watching the particle, but from time to time I'm applying a field that takes the particle. This is a charged particle, and I'm applying a field that takes the particle to the origin. And I'm asking, will it help or not? Fine, but there are other situations. If you're considering these computer algorithms, stochastic computer algorithms, sometimes you don't know where are you in the phase space. Are you close to the solution or far away from the solution? You have absolutely no idea. If you knew, you would just go to the solution. So people apply resetting because they